Hello, my name is Riley Gillis. I am the founder and director of the Nevada Medical Research Consortium at the University of Nevada, Reno. Go Pack. Today's topic is journal clubs. This is an updated overview as of spring 2025 on what you should expect at NMRC journal clubs and what you should prepare for when it comes to you presenting at these presentation opportunities. Without further ado, I'm going to share my screen and let's get into it. At the conclusion of this presentation, I want you to understand what a journal club is and why they are important. I also want you to understand what NMRC requires at our journal clubs and view an example journal club presentation. The mission of the Nevada Medical Research Consortium is to generate longitudinal mentorship for researchers, foster interdisciplinary collaboration between laboratories, and ultimately create informed medical scientists for Nevada. In response to objective number 3, we have implemented journal clubs. This is a definition taken directly from our Constitution, which you can view online at nevadamrc.org. Journal clubs are composed of individuals from a specific scientific field who meet periodically to evaluate novel scientific discoveries and advancements. Medical and scientific literature will be reviewed to create informed medical scientists familiar with the most recent discoveries in their field. Basically, you will choose a journal article that was published somewhat recently within the past few years, and you will present on it to your department. The requirements are quite simple for NMRC. Departments will meet at least once per month for journal clubs and general affairs. Student researchers will take turns presenting on new scientific articles pertaining to their field of investigation. It doesn't have to directly pertain, but if you think that it is something that will be useful for you to learn about and for you to teach others in your department about, then that is a highly encouraged article to choose. Each researcher is expected to participate, asking questions, giving comment, and presenting periodically. You will need to consult your department chair for department rules on required presentation frequency, as some departments are much larger than others. And so the frequency at which the members will present is going to contrast there. So just discuss with your department chair. Informal presentations, keyword, informal. You're not defending a doctoral thesis or a dissertation here. Okay, there are informal presentations. We understand that you are a student with a full class load, and we don't want to be adding more stress to your load. This is something that is here to benefit you. And so presentations are informal, as you'll see today with my presentation. But they do need to include, nonetheless, an introduction to the field. Not everybody in your department is going to be specifically familiar with your field. They might be more broadly familiar with your work and your field. And so you do need to have an introduction for them so that they're not lost. And then you also need to dissect the chosen scientific article. You need to discuss their methods, their results, their own discussion. And I want you to point out what was quote unquote good science. Okay, so maybe they had a very high sample size, or their statistical analysis techniques were led to much reduced bias over other statistical analysis techniques or something like that. Those would all be good things to point out. And I also want you to point out what could be improved. So perhaps they published in a really low impact journal, or they obviously cherry picked data or used a very small sample size. These would all be good discussion points. I also want you to connect the scientific article to your own research and or to implications for your field. And you do need to have citations. I would encourage you to pursue citations that are, kind of generally accepted by your field. So if you're a psychologist, then use APA. If you're a microbiologist, you might use ASM, for example. Here are the benefits of Journal Club. You will gain valuable presentation experience. This is something that not a lot of undergraduates get to have, and I think it kind of shocks them when they get to graduate level journal clubs, presenting at national conferences, interviewing, and even presenting their thesis. I've met plenty of graduate students who, when they defended their dissertation, were super duper stressed out, more than the normal amount of stress that you would feel for something like that, simply because they did not get comfortable with presentations early on. And so I would encourage you to participate in this, give it your best shot. Oral presentations are something that are not going to get easier unless you start doing them more. And this is a great place that is low stress. It is low intensity. If you don't do super well on your first journal club presentation, that's okay, right? It's not something that is going to really reflect poorly on you. And so this is a great place to get those practice skills so that when it does matter, you can give a convincing presentation. You'll also practice literature review skills. You'll get better at reading scientific articles. You'll be more efficient at reading them and summarizing the major characteristics of them. You'll also be able to learn and teach about your field. Staying up to date on recent scientific advancements is critical to success as a researcher or as a clinician. Additionally, passions surrounding scientific niches are intriguing in interviews and applications. If you go into a medical school interview or a graduate PhD program interview and you are able to, cite literature of recently published articles, and connect that to your thought process and your views on the world, that is going to be, I think, very impressive to the individuals who are interviewing you. It shows that you actually have a passion for what you're talking about. You're not just checking a box. It shows that you're going to stay up to date as a working professional. You're not going to stop learning just because you're not applying to anything anymore. So I think that is going to be a very, very high point for you in your applications for certain things. And then additionally, most, if not all, graduate research programs require journal clubs, and many medical schools are starting to require them. So this will typically be in the form of like a 1 credit class that you'll take maybe for 4 semesters or something like that, where you have to go and it's literally just a journal club like we are practicing here at NMRC. And so if you have this background, it's going to show the people that are interviewing you for these programs that you are prepared to take on that responsibility. Then it's also good for your own comfort. Yeah, so you'll excel in those journal clubs. Next, it will add scientific presentations to your CV and resume. This is just another place where you can start adding stuff to your resume. You don't have to go to a national conference to get presentation experience. You can totally add these presentations under your presentations section of your CV. You'll also develop relationships with department members and network. For example, you might find somebody that is researching something that is tangentially related to what you're researching, and perhaps you'll develop a valuable relationship with that person over Journal Club, and maybe that will be what fosters the interdisciplinary collaboration. Finally, you'll begin to understand what factors make some articles more or less likely to be accepted for publication. In other words, what should you be applying in your own research as you start to publish articles? This point down here, the what is good science, what could be improved, is major for that. If you're reading articles and you're starting to identify, oh man, that was kind of a cruddy article, or that was one of the best articles I've ever read, and you're able to tell why, then you'll be able to employ those in your own publication endeavors. Okay, let's get into an example journal club presentation. I'm going to stop here and I will catch you on the flip side. Okay, let's get into an example journal club presentation. Hello, my name is Riley Gillis. I am a research fellow at the University of Nevada, Reno School of Medicine in the departments of internal medicine and microbiology and immunology. My primary research goals are to develop rapid diagnostic tests for the point of care detection of various infectious diseases. This includes everything from COVID-19 and Ebola to biothreats like Eastern equine encephalitis virus that are termed Category A weapons of mass destruction. Today, we will be discussing field performance of 3 Ebola rapid tests. This article, published in Lancet Infectious Diseases in 2022 by Daniel Mukadi Bamuleka et al., is titled Field Performance of 3 Ebola Rapid Diagnostic Tests During the 2018 through 2020 Outbreak in the Eastern Democratic Republic of the Congo, a retrospective multi-center observational study. So these researchers were looking to evaluate 3 new rapid tests for the diagnosis of Ebola in clinical settings. Ebola was first identified in 1976. It's a disease that is caused by 4 distinct viruses from the Filoviridae family. It is a hemorrhagic fever, meaning that a primary distinguishable characteristic of the symptoms or signs is uncontrollable bleeding, both internal and external. The virus causes systemic inflammation, which damages tissues, causing them to leak. It targets clotting factors, causing uncontrollable bleeding, internal and external. And patients ultimately die of blood loss, dehydration, shock associated with blood loss, etc. It is endemic to resource-limited regions of sub-Saharan Africa, primarily occurs in places like the Democratic Republic of the Congo, for example. It kills up to 90% of those infected. So by the numbers, it's roughly 90 times deadlier than COVID-19. There are no treatments. Current treatments focus on supportive care. Okay, so this is giving people fluids, giving them electrolytes, anticonvulsants, stuff to help with blood clotting, but no real treatments there. There is one vaccine, but it only works for one species of Ebola. And like I said, there are 4 distinct species that cause Ebola. So there are 4 viruses. This only works for one of the viruses. If you contract one of the other viruses, then this vaccine is going to be rather ineffective for preventing disease. It's spread by bats, monkeys, humans, through contaminated bodily fluids like blood. And it is highly contagious. Now, this study was looking at evaluating lateral flow immunoassays, which is the technical term for a rapid test or a rapid antigen test. This is everything from the COVID-19 rapid test, which I'm sure we are all dreadfully familiar with, to pregnancy tests. They actually function the same way. Rapid diagnostic tests use antibody-antigen complexes to reveal lines. Antigens are biomarkers of disease, so typically a surface protein or glycoprotein on the surface of a virion or bacterium that your body can use to identify a pathogen. Antibodies are how our body identifies pathogens. And so it's a part of the adaptive immune response. It's a soluble protein that our body secretes that binds to specific antigens. Okay, and when they bind to antigens, it can neutralize the function of that antigen. So maybe that antigen was a glycoprotein that the virus used to gain entry into cells. And then it can also essentially mark that pathogen for death. So your other immune molecules in your body will notice that something is marked by an antigen, by an antibody, and go to hopefully try and destroy it. For a rapid diagnostic test, you'll notice that 2 lines means you're positive and 1 line means you're negative. So this is a positive pregnancy test. This is a negative COVID-19 test. Rapid diagnostic tests are much more accessible than traditional diagnostic methods. They're super cheap to produce. They are easy to disseminate. In fact, the United States sent COVID tests to practically every single household in the United States during the pandemic, and that is not something that would be feasible for other diagnostic tests, like, for instance, PCR or an MRI machine, right? That's a diagnostic machine. So they are easy to disseminate, and they often don't require laboratory infrastructure or training to use. The average person, the layman, can use a rapid test, no problem. And they can diagnose at the point of care, meaning somebody can go into a clinic and get results that day, or take the test home, get the results within 20 minutes, which is critical to treatment outcomes, right? So somebody has right, non-specific symptoms. They come in with a headache, right? You don't know if they have a cold or an allergy or if they're dehydrated or maybe they have Ebola. Accurate diagnosis will inform your treatment of that individual. It will also inform prognosis, and very importantly, it will inform biocontainment. So if you're trying to control an outbreak and you're able to identify who has this disease and who does not have this disease, then you're going to be able to accurately quarantine people. But there are some challenges to rapid tests. Historically, they are less sensitive and specific than PCR or culturing. So they're known to have more false positives and more false negatives than molecular tests like PCR. And there are gaps in the diagnosis for Ebola, as we will get into today. So how does a rapid test work? There are four major parts. There's a specimen that you're going to add to it. This is an analyte, right? So something with human blood or serum or urine or spit, something that is going to contain those antigens of interest, right? Because it has that pathogen. There's going to be a conjugation pad, and then a test line and a control line. Now, as I said before, 2 lines means that you're positive. If you take the test and 2 lines show up, then you are positive for that antigen. If 1 line shows up, then you are negative for that antigen. And typically that's going to correlate with if you have the disease or not. If there are no lines visible, in other words, the control line is not visible, then you have a defective test. That's why the control line is there to tell us if the test is working or not. Here's a schematic of a rapid test so you can see what's happening on a microscopic level. In the first stage of a rapid test, you will add a sample to the sample pad. Okay, so here's a drop of saliva, let's say, and these are pathogens, and you'll see these little spikes on the pathogens are the antigen. Okay, that's the antigen of interest. And then there's a conjugate pad. This conjugate pad is going to have a number of antibodies, which, if you'll recall, bind to antigens. When you add this analyte to the pad here, it's going to wick across the entire lateral flow immunoassay. It's called capillary flow. It's the same way that if you get a paper towel wet, the water will spread. So in stage 1, you will add the antigen, right? And the antigen will bind to the antibodies, which will be visible. Okay, so these will be faintly visible. And if they congregate at a certain section of the pad, then you'll see a line appear. So as this antigen binds to these antibodies and the capillary flow is activated, it starts wicking up the pad. It's going to carry those antigens across, and the other side of the antigen is going to bind to these antibodies. Now, these antibodies are not visible to the naked eye, but since they're sandwiching this antigen, essentially gluing these 2 antibodies together, then you'll see a line appear at these lines. Okay, so a line will appear here at the test line if you have that antigen. And at the control line, these are antibodies that simply bind to other antibodies. So, for example, these antibodies at the conjugate pad are going to wick across the lateral flow immunoassay if there's antigen or even if there isn't antigen. And so unbound antibody here is going to be immobilized here, and you're going to see a control line appear. So in other words, you'll add a specimen, it'll carry some colored antibodies up, they'll assemble on a test line, you'll see a line appear if you're positive. No matter what, they should be assembling on the control line. So a line should always appear there. So let's get into this study. This study used banked blood samples from Ebola patients obtained and tested retroactively throughout several different outbreaks. They used RT-PCR, so reverse transcriptase polymerase chain reaction, which is a definitive diagnostic technique. If you'll recall, I mentioned earlier that it's much more specific and sensitive than rapid tests. So they use that as their control, if you want to call it that, versus the 3 rapid tests, which will be the test. The 3 rapid tests they chose to evaluate were the Quick Navi Ebola system, the OraQuick rapid test, and the Chorus Ebola antigen KSET rapid tests. They did do statistical analysis. They compared sensitivities. Okay. So the sensitivity refers to how sensitive a diagnostic is in response to the presence of the target detection marker. So in other words, if you have a very, very, very small amount of pathogen, is it going to detect it as positive? It should if it's very sensitive. The percent sensitivity is calculated from the proportion of true positives from the rapid test out of all positives in the tested population. Then they also compared specificity, which is effectively the opposite of sensitivity. Specificity in a diagnostic test refers to the intervention's ability to accurately display a negative diagnosis based on the lack of identified detection markers. So in other words, if you don't have the disease, you should never test positive on one of these tests. And if it has high specificity, then you will not be testing positive. You will not be getting false positives. Percent specificity is measured by the number of true negatives from the rapid test out of all negatives in the tested population. So here is a schematic from their paper showing how they collected samples. So they had over 9,000 potentially eligible samples. Remember, these are banked blood samples from previous Ebola patients. They excluded 166 samples, including 146 samples from deceased patients, and 20 samples from a site where the test was used less than 20 times. So they wanted to make sure that there wasn't any bias in these results because of really low sample size in some of the studies. So that left them with a little over 9,000 eligible samples, of which about 7,500 were tested with the Quick Navi Ebola system. About 1,500 were tested with the OraQuick Ebola system, and less than 1,000 were tested with the Chorus Ebola antigen K-set system. Now, if we follow this flow chart down, let's just go to the Quick Navi section. You'll see that 7,000 of the 7,500 samples tested were negative. But of those 7,000 that tested negative by this system, 87 were actually positive. So Gene Expert is an RT-PCR system, and so it is a definitive diagnostic technique. So that is not super duper great, right? Because we would want there to be zero false negatives, right? But here they're saying that out of 7,000 samples that they thought were negative, in fact, 87 were positive for Ebola based off of this test. Conversely, if we look at the positive tests, only about 500 of the samples were positive. And of those positive samples, 25 were actually negative. So there were 25 false positives. So ideally, we would want there to be zero false negatives, zero false positives. And the same is done for each of the subsequent tests. Okay, so here you'll see a figure from their paper that they published. It's looking at sensitivity versus the cycle threshold value on the x-axis. The cycle threshold value correlates with how much nucleic acid is present in the nucleic acid amplification during PCR. So in other words, over here, there's going to be, very low pathogen, like low amounts of pathogen. Up here, there's going to be much higher amounts of pathogen. And we want the sensitivity to be, in an ideal world, it would be at 100% across the board. But we don't live in an ideal world. The Quick Navi Ebola system had the highest sensitivity, followed by the OraQuick Ebola system, and finally the Chorus Ebola antigen K-set system down there at the bottom. They concluded that no test approaches World Health Organization standards for sensitivity. In 2014, WHO published a target product profile for point-of-care rapid tests for Ebola virus disease, stipulating a desired clinical sensitivity of at least 98% and a specificity of at least 99%. When we look at these specificity percentages reported from the data in this study for these three rapid tests, they're all getting pretty darn close to that 99% minimum, which is a good thing to see. One test, the Quick Navi Ebola system, actually exceeded that 99% desired minimum. So healthcare providers can largely rest assured that if somebody tests positive on these three rapid tests, they likely do in fact have Ebola, should receive treatment for Ebola and not be sent home. There's a very low chance, given this data, that somebody's going to be getting a false positive for Ebola. Now, when we go to sensitivity, that's a different story. The highest sensitivity, also the quick Navi, was only 87.4% with these kind of broad confidence intervals. So it could be as high as 96.8%. But they're saying 87.4%, which is rather low, and 38.9% for the chorus Ebola antigen KSAT rapid test, which is incredibly low. What a 39% roughly sensitivity would mean is let's say we have 100 people with Ebola, and we test them with the chorus Ebola antigen KSAT rapid test. Only 39 of those results would come back as positive, which means that perhaps the other 61 people would be sent home and would suffer with Ebola and perhaps give it to their family and their community. So that would be a lot of false negatives, a lot of diagnostic escape, and a nightmare to try and control an outbreak of a deadly infectious disease. Given this data, I would say that perhaps these Ebola tests are good screening tools. So if somebody, that's at a higher risk for Ebola, tests positive, you can rest assured, okay, this is an individual that we need to quarantine, but you should not be sending people home because they tested negative, because you don't know if they have Ebola or if they don't have Ebola in this case. They said ideally, rapid tests would be used to triage and rule out Ebola virus disease, requiring high sensitivity but potentially accepting a lower specificity among people with clinically suspected Ebola virus disease at the peripheral care level. None of the evaluated rapid tests fulfilled these criteria in their study. However, the available rapid tests could help to sort people with a high risk of suspected Ebola virus disease from people with a low risk of suspected Ebola virus disease while waiting for confirmatory gene expert, which is PCR, Ebola assay reference testing at the triage site. So in other words, what they're saying is in a pinch, they will be using or they will condone the use of these rapid tests, right, to screen people for Ebola, but that it should not be used as a definitive way to rule out having Ebola. So in other words, if someone gets a negative on the rapid test, you should probably wait to send them home until you can confirm that with another diagnostic technique, such as the gene expert RT-PCR Ebola rapid test or molecular test. Because yeah, you'll see here, right, 38%, that is really, really low. So yeah, so let's talk about some strengths and limitations of this study. They had a lot of samples. That is a huge strength. They included comprehensive inclusion and exclusion criteria. So if we go back to here, right, they had written out what is going to disqualify a sample from our study. Why are we going to include these samples? That's always an important thing to consider. They did include 95% confidence intervals for stat analysis. So they did find that their results were statistically significant. You'll see that here, right? Dashed lines, they even included the 95% confidence intervals in this graph. They unpacked which antigens were used. I know I didn't really talk about that too much, but with a pathogen, there are, perhaps hundreds of different antigens that you can choose from when developing antibodies for a rapid test. And so they were able to talk about which antigens the tests were actually targeting and detecting. And I think that is very clinically relevant as we look for, what is the abundance of these biomarkers at different points in the natural progression of the disease. They also discussed clinical implications. They didn't just stop at saying, okay, here's our data. They said, here's how we should be using the data. We should be using these to screen for Ebola, but not to definitively rule out Ebola. And also the Lancet, which is where it was published, is a very high impact journal. And so they have a wonderful history of really critical peer review. And so we know that this study can be trusted. Here were some limitations that I found. There were disproportionate sample testing. And so you'll see here in the sample selection, right, there were 7.5 thousand tested with the Quick Navi and only 845 tested with the Chorus Ebola antigen test, right? And then the Quick Navi scored the best and the Chorus scored the worst. And so 800 is still, a considerable sample size, but this is much bigger, and it would have been nice to see that more homogeneously distributed across the different tests. Just because I wonder, maybe the Chorus isn't actually this poorly sensitive. Maybe it was just the lower sample size, but you never know, right? Until you do the study with a large sample size. There were no discussion of applicable species. And so if you'll remember in my introduction, we talked about how there are 4 different viruses that can cause Ebola. And this paper didn't address the different viruses. So I know from my own research, right, that certain rapid tests are only sensitive to specific species of the virus. So, for example, the Quick Navi might only be sensitive to the Zaire Ebola virus and not the Sudan Ebola virus. So that's another place where diagnostic escape could occur. And there was no discussion of limit of detection. So this is the absolute least amount of pathogen that you need for something to test positive on a test. I would have liked to see that. So there do remain gaps in rapid tests for Ebola. They are still useful screening tools, but Ebola cannot be ruled out with these 3 rapid tests alone. There remains a critical need for commercially available rapid tests that do meet the WHO standards, and commercially available pan-reactive rapid tests. So this is a rapid test that can detect all species of Ebola, which I've looked into it, right? And I know from my research that none of them can detect all of the species at once, which is unfortunate because somebody comes into the clinic with Ebola, you might have to give them 2 or even 3 rapid tests in order for them to test positive. That's inconvenient, time-consuming, mistake-conducive, etc. There's also no commercially available rapid test for Thai forest Ebola virus, which is one of those 4 species that causes Ebola. This is the goal of the Equine Laboratory, the second point, not the third point, to develop commercially available pan-reactive rapid tests. So hopefully we will have a test where you can just take one test and know if you have Ebola or not. There are also discrepancies in manufacturer-provided data versus empirical data. And so you'll see in the Daniel Mukadi Bamaleka et al. Studies, right, like the one that we looked at today, that the manufacturer might report, oh, we have a 98% sensitive test, when in fact, they have something more like a 38.9% sensitive test, which is a substantial discrepancy. And that's one of the reasons that these comparative studies are so useful. It really holds people accountable, Okay, this is the end of the presentation. At this point, you would want to take questions from people, perhaps have a discussion with people that are interested in your research. Hopefully, people will have learned something from your presentation. You would have gained a greater understanding of your field from your journal club presentation. That's going to be it for this video. Thank you for sticking along. If you have any questions, please reach out to NMRC at med.unr.edu, or you can reach out to myself at rgillis@med.unr.edu. And we will get back to you. Your department chair will also have good resources. We have released some kind of guidelines or rubrics that can make it easier to follow as you start making your first presentations. So yeah, good luck. Try not to stress over it. Have fun and get out there and learn something. Thank you.